Good afternoon, everybody. I'm Jim Spencer from the School of Cellular and Molecular Medicine. Most of what I do is related to antimicrobial resistance in bacteria. In this context, I want to talk about efforts to develop novel antibacterials, specifically to understand the mode of action of novel antibacterials, so how they kill bacterial cells. The approach that we're using is an image analysis-based approach developed by collaborators called bacterial cytological profiling. So originally around 10 years ago, what they did was label bacterial cells, challenge them with antibiotics, and use principal component analysis to classify morphological responses to agents with different modes of killing, and use that to understand killing by unknown agents. That was then. What our collaborators, who do all the heavy lifting, we do the image acquisition, are now doing is extending this to the single cell level using deep learning approaches. So we label bacteria in the presence of different antibacterial agents. We measure around 60 morphological features and use deep learning then to classify these features and compare with our unknown agent here. So what we can see is that our investigational compound is doing things to bacteria that are different to known agents.